It actually has it's a synthetic embryo.
Right. So this is synthetic seed. This isn't synthetic seed.
I guess,
this is your kind of version of it a little bit.
Yeah, we can do both, but with an embryo, you can actually get root and shoot circle just like a seed.
Sure.
Right.
Mm-hmm.
So picture planting, sour diesel that's true to type.
Right.
Basically a clone from seed form.
Yeah.
Does it make sense though?
There you go. There's a shot of it, folks.
Yeah.
There's a shot.
Oh, those things are.
We're Jug Dealers, we're the podcast you need in your feed. Teaching the game like you never seen. Jerin, Gabe, GRAMBO, true OGs. Masters of the globe, set your mind free. From Denver, Colorado, where the data's green. We set the data points for the SOPs. Jug Dealers Podcast, yes indeed.
Hey, everybody. Welcome back to the Jug Dealers Podcast, where we discuss all things cannabis and lifestyle related, brought to you by the good people at 5-8. And as always, available at jugdealers.net and jugdealers.shop. Jayhawk, we're back in studio today.
Yes, we are. Also, always remember you can request samples at jugdealers.net/samples. We also have a bookings page where you can, there we go. We got the growers form. Fill out the growers form. Yep. We've got 3 different packages. If you're a commercial person or working in a commercial facility and would like, something else, you can also go to the comment section and just shoot me a direct message and I will deal with that. Also, we've got a bookings page if you want to request our presence anywhere, like at a cup or at an event.
We went and visited the boys in South Dakota.
Yes, we did. Shout out to Bad River Cannabis. Thank you guys so much for having us. That was a heck of a time. It was quite the drive up there, but thank you for showing us the hospitality and honestly, great work with what you guys are doing. I was impressed with the quality of the flower that you guys gave us.
What they did with that garden was amazing. They really
keep up the good work. Keep it up. I see bright things in your guys' future. But without further ado,
we got a special one today.
We really do have a special one today. And, as promised, as we've been continuing to promise, we're going to bring in, other guests on specialized topics that quite frankly, know a lot more than we do. We like to introduce a lot of different topics and talk about what we know about it.
Well, and this is one that's hot in the industry. It has been for a couple of years, but the reality is people have been doing it in other industries for a really long time. Yep. We're just getting it in our industry. But I think one of the things that's pretty unique is, when you talk about strawberries, people are only growing one kind of strawberry.
Sure.
And we're doing all kinds of stuff. So that's why we really found it necessary to bring this guest in, because he's going to put to bed some myths and just, give us some information that I don't think a lot of people are putting out there.
Well, and I think you guys are going to be able to, shut down the computer, shut the laptop, whatever, and actually go into your grow room and have some really practical information to leave with, which is one of our goals on every one of these shows. So anyhow, without further ado, we are going to step into the lab with our good friend Jesse, who is the director of Mothership Labs. We're going to talk about the next frontier of cannabis propagation, which is, tissue culture, which also has to do with, of course, long-term genetic preservation, and also synthetic seeds, which when we were in the lab, that was...
when he showed us the synthetic
really
seeds,
interesting.
It was like, whoa, this is next level shit.
Well, and I would say, if you're really wondering what's actually possible and where things are going, with whether it's synthetic seed and plant cell manipulation, things like that, really this show, truly is for you. So again, here is Jesse with Mothership Labs. And why don't you introduce yourself and maybe you give us just a brief background on where you came from, and then really what your goals are. And then we'll start getting into the meat of this thing.
Yeah, thanks for having me, guys. Basically started in the cannabis industry. So I was growing, managing facilities, everything. And then I went to biotech for a little while. DNA, learned all kinds of stuff. And then came back to the cannabis industry. So we built a lab and did research for 4 or 5 years. Hard research. It was a long 4 or 5 years.
Okay.
And
what were you working with in Big Ag?
So we were running tests, basically looking at certifying seed stock for different breeders. So
gotcha.
In order to sell seed on the market, you have to, get hybridity testing. So DNA work, bacteria, viruses, make sure they're clean. So farmers start growing them and destroy their crops. Right. So,
right. And obviously, you know, some of the things that people think about, you know, when it comes to tissue culture in particular, as Gabe already mentioned with strawberries, you know, that's a particular plant that's very virus riddled. So obviously people are buying clean stock, but that's far from the only, you know, place that kind of technology is, you know, is implemented. And of course, there's also, you know, a difference between preservation and cleaning and things like that we're really going to, you know, let you get into a bit deeper. GRAMBO, can you move that up a bit? There we go. All right. So
a little bit more.
Let's I mean, quite frankly, let's just really define really what tissue culture is. And why don't you give like your kind of running and evolving definition, which I'm sure it kind of is, of tissue culture.
Yeah. So tissue culture can be used for a ton of different things, right? What people think of today, especially the cannabis industry, is like you can clean genetics, you can store them and propagate. But you can do a lot of different things, genetic modification, research. You can do, I mean, there's a lot of different applications.
So, and also like identifying genetic markers, things like that, right? So even for, I mean, because of course, you know, it's one thing when you're, you know, a lot of times in the ag world working with, you know, one particular strain or maybe a small group of strains as opposed to, you know, in theory, hundreds of different ones, but in reality, it's probably like, I don't know, 5 to 7, in some way, which, you know, and I don't know, I want to get into that a little bit later, just about, you know, really where is like genetics at this point, you know, because you've probably been a person that's had you know, an ability to take a different look at that, as opposed to just trying to match names with names and match their breeding history. You're actually going to be looking at it from more of a genetic level, molecular level, which is not something we've had a whole lot of time to do with cannabis, right? I mean, this is, you know, cannabis is still really young, and especially an ability to do any kind of research. I mean, what really made, you know, you and your team and you want to get back into, or even just get into tissue culture in the first place? Is this something you kind of fell into? I mean, just from like, what is your educational background? Really, how did you get here?
I look at it more of like a tool, right? Because I've always been breeding and growing I mean, since I was young. So the more I learned, the more I realized it's like I wouldn't say a problem, but the room for improvement, especially for tissue culture and genetics in general. So it's kind of where we shifted the front facing part of our company. Yeah. You know, we've been doing it for a long time, but you know, we kind of came out of research and it was a hot topic, and we're like, there's not a lot of people doing it well, at least at the point when we were coming out. So focused on developing it so it could be useful. Storing genetics so they didn't mutate and have, you know, a bunch of problems pulling them out of tissue culture. One for research, breeding, you know, keeping your phenotypes on a shelf until you flower them out, selection, just overall efficiency. You can use tons of applications for that stuff. So while we built the lab and we were, you know, in the research part, it was just another tool. We were looking at population genetic stuff, doing, you know, developing molecular markers and screening tons and tons of seed lines.
When you say molecular markers, kind of go into that a little bit as far as for our listeners, as far as like, what does that really mean? Does that mean like how tall it's going to be, the size of the leaves, THC? Like, what is that? What are you looking for?
Exactly. Yeah. So,
okay.
Well, I guess that wraps it up.
Good talk, boys.
Good talk. Yeah. So if you think of a molecular marker, it's not like one molecular marker for whatever, right? There's a lot of things you can use as a marker. And what it is just a pattern of, you know, DNA or.
Okay. Yeah. That was going to be, are you looking literally at the helix of the chain and being like, hey, this says it's going to be tall. This says it's going to have long internodal spacing. This says it's going to be. Now, riddle me this. Is this the type of thing where, you know, you can still screw it up? Like this is like, this is like maximum potential. Like, you know what I mean? Like when you look at these markers and it says it's going to be, you know, 30%, like it could potentially be 30%, but if you don't grow it right, it still could be 20, you know?
Yeah. Cause you know, once you identify the genes, it's genotype by environmental interaction.
Mm-hmm. We say that all the time. Part. Yep. That's phenotype by definition.
Yeah. And of course there's always genetic potential, right? So even if you have the genetic potential and, of course, when you're looking at sequences of genes, which, is a little bit, I think, probably more broad than being able to say like super specificity to, but I do find it interesting to really be using that to look at, and some people have kind of started that, and I don't even want to use their name, but there was a company that was collecting a bunch of different things and doing different genetic markers and such.
So
it was interesting the premise and what they were trying to do in the sense of like, let's look at what genetics we really have and how much has the gene pool been narrowed and how much of that has been lost, and do we need to go back into some of these essentially these places of origin to try and regain some of the genetics. Hey, everybody. Didn't want to interrupt the flow of the conversation, but did want to remind you that anything we talk about that are our products, you can find at jugdealers.shop 24/7 online. Go and get it. Support us. Help us continue doing what we're doing. Jugdealers.shop. Thanks a bunch. Now back to the show. Essentially these places of origin to try and regain some of the genetics.
Well, what stage do they need to be at for you to get the genetic markers? Do they have to be can they still be in seed, or do they need to be grown out a couple weeks? When can you take that?
Pretty much any time you can get DNA, right? So if you're getting it from seed, that's destructive. So you're not going to be able to pop that seed and see what it is.
Okay.
Right?
Okay.
But the idea is, let's say you've identified molecular markers. So the example I use is like the color purple, which is easy to understand, right? So we know what genes cause purple. You can have the parents, cross the parents, make the seed, and then plant the seed. And when they're, an inch or two tall, that first set of leaves, you can take about a hole punch size, extract the DNA, and then test all the seeds, just the flattest seeds, however many you want.
Sure.
If they contain that genetic marker, then it's going to be purple.
Right.
And you can select there. So kill if you're breeding for purple.
Right. If that was it, just, again, watering it down, but it's just very simplistic. Yep. Using something to really try and go in that direction. And I mean, obviously that's going to be one of the, in overall, and as an overarching thing, because of course my mind's going all over the place, just those benefits of tissue culture, which seems to be vast because of the manipulation you can do as opposed to traditional cloning, right? Because obviously traditional cloning, you're taking the genes, the genotype that you get wherever you got them, whether you got them in Oklahoma or you got them in Florida, we bring them back to Colorado, and then we're going to see how that phenotype kind of works out. Sometimes it works out well, sometimes it doesn't. But, the cloning thing, even though, and we've always said, and Gabe and I have talked about this a lot. I mean, obviously popping seeds is great, and popping seeds in your own environment, so you're selecting for your own environment, is fantastic. But there's still a sifting process with clones. People think that's such a shortcut.
I think that's one of the beautiful things about what you're talking about, because I am someone who's put down, I think the most seeds I've ever put down was 500. And, I always tell people what you're looking like. I respect the fact that people go out and they buy a 10 pack for $100, $200, and they want to find something in that 10 pack. But I always tell people, what you're going to pick out of 10 is completely different than what you're going to pick out of 500, and if there's a way to kind of circumnavigate, hey, going through the 400 that we all know are, I'm not saying they're not good, but they're really similar. And I think for people like us, we're looking for the weird stuff.
You want the outliers.
I'm looking for the outliers. I'll never forget with Chem 95, the one that I found that was the one I really wanted had long internodal spacing, grew like OG, smelled like Chem, and out of 500 seeds, there was freaking one, so to be able to identify.
At least there was one.
Yeah. And at least there was one. God knows what happened to it. What would you say are some of the biggest benefits as far as tissue culture versus traditional cloning, just for our listeners? Because, I think a lot of people look at it, and I can tell you from experience in these big commercial buildings, what they dump into some of these labs is quite a bit of money, and then I see you kind of doing tissue culture at DGC, out in the open, and you're doing it pretty cheap and pretty effectively. And I think, as we get into this episode, we'll talk about it, but some of the services you offer and some of the processes you offer are really cool. But when you break it down like that, what are some of the benefits you see over tissue culture versus traditional cloning?
I think the first one disease, pests, all that stuff.
Yeah, obviously.
I'm still getting clones in that covered in mites, or I've seen it all, right? So the first benefit, getting, having clean stock. All the clones that I work with, or genetics and just everything I work with, I put it through TC because it's not going to have anything coming out, right? A lot of these pathogens spread rapidly. Everybody has something.
Of course, which is also why you're doing tip meristem, right? Because if you were just storing genetics, and is that something that you do? Do you do nodal cutting as well, just for preservation aspect, or are you always just from an SOP, always doing tip meristem?
I always do, meristem culture on everything.
Yeah.
It's, viruses,
of course,
just everything.
Right.
For the listener, can you just explain the difference between the two? Because I think it's a little bit complicated for people who don't, there's a lot to it.
Yeah. So node culture, which I see a lot of people doing, you're taking, a little chunk of stem right, with the node on it. And you might be able to get rid of a lot of things. Part of the process is you're surface sterilizing, right? You're not completely frying every piece of that tissue, right? So if you have something left in there, it's going to grow back. You might not see it for a while. That's why I start with meristem tip culture. So the difference there at the very, very tip, right? These new cells that are rapidly dividing and
the meristematic dome.
The meristematic dome is building new tissue, right? It's going to be a stem or leaf or whatever.
Yes, sir.
Once it's tissue, then it's connected to the vascular system, and these pathogens can get in there. So that's the basis of essentially it's mechanical removal of this chunk of clean cells that don't know what they're going to be yet.
Yep.
So if you remove that, then there's no viruses or pathogens that can sustain in there.
And I know one of the reasons that people will see some of these kits out there where you're doing nodal cuttings of it. I mean, that's obviously a lot easier to do. You're not having to peel all the way into the true dome to get those last couple cells, which are basically clear.
I mean,
we've got some cool videos.
It's like a grain of sand. If you've watched Jesse do it, he's got a video screen and he's really doing it, looking at the screen, and you got it down to like a grain of sand, basically.
Fortunately, we've got some wonderful 4K video that we will be putting up there because we we did about a 45-minute video with him walking through all. So we're definitely going to be putting clips of that in here as well. Hey. So that is one of the cool things is we're going to be able to actually, turn around and show you guys some really good video of everything we're, talking about. But I only want to make that point is there's nothing wrong with nodal cuttings.
You make that point a lot with people because
and it's good for storage and you're easier to do.
Right.
And for surface sterilization, things like that. But for getting rid of actual viruses, that's not going to cut it. But again, let's say you did have some plants that were riddled with different bugs and things like that. You would get that surface sterilization and that would actually be of some benefit. So I'm glad he actually said that. It's not that it's no benefit. And for genetic storage, it can be great. But I also, one of my biggest things is, and we talked about it a little bit when we were at the lab, but, truly is that genotype, plus environment and phenotype. So what happens even when you have some of these genetics? Let's use GG4 as an example, right? One of my favorite all-time genetics. Everybody on here knows actually is, that's my favorite flower. But that was the first place anybody saw dudding, right? And I remember I want to say it was on the Roll It Up forum. I want to say it might have even Cap that dubbed it dudding. And it's the only plant that I've truly ever seen it on. And it's a single, whether it be a single branch or I've actually seen a whole plant, right? And it comes and it goes, hence the latent aspect of it, right? So what if you take some of these things and correct me if I'm wrong, cannabis can be actually pretty good at handling viruses. It can take them, it can keep trucking along pretty good until it just gets a little bit too many monkeys on the back, and then it really starts faltering. And that's when people see that genetic drift, which I really like to say, clonal accumulation and not clonal degeneration, because I really think it's more of that tired because it's taken on so much over time. And then that's going to affect again that phenotype, that expression, right? Because that being part of the environment environment, the pests and diseases it's dealing with. So as very long, maybe somewhat roundabout question, like, are you seeing a different phenotype with some of these classic genetics that are brought in, cleaned up, and then put back out of culture and grown out? Can you speak on that a little bit? And is there any variation? And if so, is it predictable at all?
Absolutely. Yeah. We see a huge difference. So there's a lot behind it, right? So when you say genetic drift, and I hate that term.
Yeah, you're my guy. You are my guy. You are my guy. Yes, please go on, sir. Please.
So that's, for the record, that's population genetics term. Right? But what people see, right, over time, you're just cloning off of clones, off of clones. You're just accumulating, and doesn't matter where they come from, different endophytes and, fungus, bacteria, good and bad, and stress. Right, throughout, if they run through a period of stress, even if you bring them back and they're doing well. So you're getting a collection of these organisms in there, microorganisms, as well as there's a fair amount of like epigenetic stressors that start accumulating. So the main one being methylation. So it doesn't change the DNA itself, but it starts limiting its ability to express.
Okay.
Right.
Okay. Because genes can be turned on and off, right? And so it limits its ability to do that as freely as it should be able to, correct?
Absolutely. Yep. And it does it in humans too, which is terrifying.
Oh, shit.
So
it's a whole other podcast.
I'm not fully expressing myself right now.
Okay. Shit.
But yeah, that's I guess somewhat of a newer field or focus with this is epigenetics. You guys might have heard that stuff too. But what it is it's starting to limit its ability to transcribe and, the DNA itself. Right? It doesn't change it.
Sure.
So with a lot of those things happening, then over time, it's not going to express the same, right? If it's multiple genes that are involved in a specific trait, and some of them are limited,
right?
It's not going to be
right.
I've kind of said to folks, like, if you took a bad cut and that became the new mom, well, then all of a sudden everything from that going forward, and now it's funny because you make me pose the question in my head, like, is what we've been looking at for so long, have we just gotten so used to something that was wrong and that had drifted, had changed?
Well, see, that's what I'm saying.
And
that's exactly what I'm wondering.
And it's nothing.
Nothing against anything.
Well, and that's the thing is, like, I think there's a bunch of someone was having a conversation with me not too long ago about their OG Kush, and they said they showed it to someone in the OG Kush family. I'm not going to call them out because I'm not trying to cause any problems, but they
merely a story, folks.
But they said they showed it to someone in the OG Kush family, and he was like, he's like, mine looked a lot healthier than theirs. And he's like, I think they've just had it so long and it's just, progressively kind of been that way for so long that all of a sudden, it's kind of like I say with the diesel, even, like, I think what we remember, it was somewhat different back then. I'm not saying it's not the same.
Well, and I also think I think it's definitely the same plant.
I look different than I did when I was 20 years old. I do.
Yeah.
My face is fuller, you could tell I'm the same person, but if you pull up a picture of 20-year-old Gabe looks different than 50.
Well, and here's the thing. I definitely think it's different. And it's also different in the sense that, like, how we're running it, right? Being also part of the environment, being also part of the end phenotype as well, right? We talk about that too. Sour D goners are just gone from being run in peat moss under single-ended HPS with GH nutrients. You know what I mean? So in large part, that piece of the pie is gone as well. But what's that term? Was that methylation? Was that the specific term that you were saying? And
methylation's one of the main components.
Interesting. So that's a little rabbit hole for anybody that wants to kind of go down. And apparently that actually is an action that can happen in humans as well. So again, bottom line, it's, not expressing yourself fully. And we like to talk about, genetic expression and genetic potential, right? And even the fact that, we know that some of the biostimulants that we use, specifically in the New Millennium line, really help, like, bring out that genetic potential, things you may have not seen before. And it really just getting the plant to operate on a level where it can do its thing, right? Because really we're just stewards. We're plant stewards. We're plant janitors, depending on how you want to look at it. Stewards is a nicer way. Janitors is the realer way. But I find that very interesting because it was something that we kind of walked away after the lab. And I was like, well, what happens if everything is clean? Like, what's it going to be? You know what I mean? And is that going to be what we're used to it being?
Grambies, you mind sliding that up a little bit?
So I think for me, that was actually one of the biggest things that I had a question. I also figured that people would have a question on as well.
And this actually leads right into it. That's why I wanted him to shift it, because I remember this being one of the questions as far as, let's talk about the medium and the agar, because I remember, you actually were putting some stuff together while we were there. You were mixing it up fresh, and we had talked to you because we have the Rob Clark episode that Jerin did, we talked, and one of the big problems Rob had was that things don't look the same that, as they come out of tissue culture. And you had mentioned to us in the lab that a lot of it was the agar, a lot of it was the hormones that people were using.
And the ratios.
Yeah. Can you dive into that? Cause I think it's something really important, because I know a lot of the old heads, that's what they're most worried about, is if they kill something off and they gave it to you and then all of a sudden it comes out and it looks different, they're like, oh my God, you painted. It's like, I gave you this Camaro. It was blue. You painted it red. What the hell happened?
Well, and let me be like slightly more, specific on that as well too. I think that there has been, there's, in these other crops, in the horticultural world, there's absolutely SOPs that are set. There's ratios, hormonal ratios that are set to either keep them in homeostasis or to actually bring them out of culture, whatever. We're still, in my mind, must be experimenting, with that a bit. And there's no book that you can go to and refer to. So inevitably, there's been a lot of people that have tried to do tissue culture, and yes, it was tissue culture, but reality is, were they able to attain the goals that they had with said tissue culture? And when I sat down with Rob, that was one of the things that I asked him about. And he's like, I believe in the technology. He's like, the couple different projects that I've been involved with, we just weren't able to attain our goals. And I don't even know necessarily what those goals are at. And of course, I'd love for him to chat with you at a certain point, because
that's going to happen.
I believe one of the things you guys have done is made a big leap in evolution in your SOPs. So when it comes to what you're storing, it is the hormones that you're using, the ratio set hormones, but I'd love for you to talk about that a little bit.
Yeah. Think about a sick mother, right?
Yeah.
You're feeding it the wrong nutrients. You try to take a clone off that.
Yeah.
And it just goes downhill, right? It's hard to kind of pick that up and get the health back. And it's the same thing in TC, right? So when we started kind of looking around and seeing what's available out there, a lot of the stuff's just classical methodology or public research.
Sure.
And they're using same nutrients, or I guess basal salts, right? In the agar. I'll get to that in a second too. But the majority of people I talk to that are already doing TC, it's like MS, so Mirashigian Skoog, or DKW, which is Driver and Kawasaki. These are just scientists that made the formulation, and you can buy it.
Right.
And
it'll grow tissue, right? It'll grow cannabis. But
so you're saying that's just like a generic formula that they made?
Yeah.
Okay.
Yeah. They determined, and this is a long time ago, they determined like, this is what's required for life in vitro, right? In TC.
Right.
So, but if you look at the options, it's, different plants need different nutrients, period. Right. So DKW's for walnuts. It's a lot of woody culture.
Right.
We're not growing walnuts.
We ain't growing walnuts, folks. Right. We might be nuts, but we're not growing walnuts.
Yeah. So people have been successful getting them in and out of tissue culture.
Sure.
But if you think about just like the mother reference, like an old mom, you're feeding it the wrong nutrients.
Yeah.
Eventually it's going to die.
Makes complete sense.
Yeah. So, the issue in tissue culture, things are kind of more amplified as far as the problems that can occur. Mutations, you can get the methylation we were talking about earlier.
Yep.
If it's sitting in a vessel now, sterile and in the wrong nutrient formulations or wrong hormones that we're pumping, giving it, right? It comes out and it's just not the same. So that's where a lot of the people I talk to, they're like, I've tried TC. It's just not expressing the same. It's like Of course. I mean, you're putting it through some.
Tell me something I don't know.
Yeah. So that's really why when you started this company, I just built a lab behind my house. Said you guys.
True story. Been there. True story.
Crazy and
legit lab.
Yeah.
I mean, but
you'll see it. You'll see the film.
You have all the tech though, and that's really what it comes. You don't need a lot of space. I mean, but you have the tech.
Well, you have to have the technology and you have to have the know-how. And I mean, I guess that kind of goes into our next thing here a little bit too. It's like, can you give us a little bit of a sense of the recipe, what it kind of goes into it? And obviously you may very well have some things that are, specifics you don't want to talk about. So obviously we're not pressuring you on anything here, but also then sterility in the process, because that's always what people
you were cutting TC outside, bro.
I saw him cut TC on a farm, open air tent. GRAMBO was there.
Yeah, real talk.
So anyway,
absolutely,
if you can talk on that, that'd be great.
Yeah. So the components of your agar, right? Your medium, right? You have, nutrients, so basal salts, which you guys know all about, I'm sure. Hormones, cytokinins and auxins, so shoot and root growth, basically. Then you have your agar. So really think of agar as a gelling agent, right? Agar, there's also gelling gum. There's a bunch of different forms of this that make it, semi-solid.
Right, jelly-like.
So it's not just agar, right? Different densities of agar, so on and so forth, antibiotics, things like that. And those are just in every single formulation. So the balance of those is what's going to cause different morphology, different, stages of growth that you're trying to achieve. So if you look at hormones specifically, like higher amount of cytokinins, you're going to get shoot growth.
Okay.
Higher amount of auxins, you're going to get root growth.
Gotcha. Yep.
Right. So what I spent a lot of time on, just so I didn't have to manage employees and building, is just focusing on the core formula that produce the best health, the best results. Because if you're growing, there's so many references just growing the regular plant, right?
Right.
If you are, getting really good shoot growth and culture, and it's not the right balance of hormones, it's going to prevent it from rooting.
So not only will it's not that it just won't root well, it just flat out won't root.
Yeah,
that's a problem.
That's pretty intense, dude.
So more roots, more fruits, no roots, no fruits.
I mean, so in that respect, are you kind of customizing your media recipes per cultivar? I mean,
has it gotten that far yet? Okay.
No. Well, the reason I spent all this time on one formula, so because it works across the board.
Yeah, I would think you would have to. Yeah, I mean, again, and this is, I'm assuming, these are SOPs that you truly do want to be repeatable, right? I mean, I that's one of the things that, not to segue too hard here, but just, and feel free to even kind of go into some of the things that, with Mothership that you're doing, but the availability, it's obviously people bringing genetics in that they've had, get them cleaned up, but not this, I don't know. It's been kind of a stronghold takeover a lot of times when we've seen these kind of projects that have come up, if for lack of a better term. And people are afraid, right now, especially here in Colorado, man. It's like raw flower that you're not putting any kind of like other value into, like putting it into a bag or putting a brand on it or whatever. Like it's almost. I hate to even say this, but almost worthless, but bad. genetics. That's a different story, right? So,
I think a lot of people have finally, we traded genetics so openly. Jerin and I will talk all the time about how, I used to go to Way to Grow, and the boys at Way to Grow would just give me Girl Scout cookies, super silver sour diesel hay, like all these different genetics that, like, quite frankly, nowadays, most of them are gone. And if you have them, they're worth a decent amount of money. You know what I mean?
Genetics
is key. And again,
it's become very important again, and there's a lot of guys, again, you talk about the OG crew, talk about the Sour Family Farms, Sour Crew, a lot of these crews, they've held it down the whole time without tissue culture.
So, and obviously, you are, a bit genetic banking, cleanup and banking is a part of what you guys are doing, correct?
It's definitely a part of it. We're working directly with breeders too.
Yeah.
And I have my own collection and stuff I've just accumulated over time that I like, and I've bred and seeds and everything else. Right. But it is a delicate subject.
Right. Like what happens when I give it, it's like, oh, and I mean, and I'm not saying you, but this is what people think, right? Oh, I gave you my genetic and you gave it back and didn't keep anything, but it's like.
They know the Philo's horror story.
I didn't want to say it.
I mean, look, I'll call it out.
Yeah, no.
And it is what it is.
And again, but it seems like you guys are really working to be an open book on that kind of stuff. And I know it's still a work in progress. You know what I mean? And it's, first things first, obviously you got the lab set up. It's operational. You're actively cleaning genetics. But I would, that's an interesting question too. I was kind of wondering if there was a little bit more cultivar to cultivar, and I don't like using the term, indica sativa, but like, or, some, wider leaf varieties, like one, but again, you've kind of found one that just goes right down the middle and keeps that vigor and keeps them ready to rock.
And with what we saw at the lab, I wasn't sure because, you pretty much give your customers the ability. I mean, when we really start to peel the onion and get into some of the services you offer, you give people the ability to, with your little seed things, we'll talk about that in a little bit, but you actually give people the ability to start stuff on their own. We'll send it back to you, and by all means, in which you'll send them the stuff for the agar. So that's where I wasn't sure if there was like a certain ratio. If there was any specificity, but obviously, between that and, he's got these cool little cups. Why don't we talk about those? Cause they're really neat. You actually developed a little cup that's it's sealed top. It looks like a little shot glass. It's plastic. And then in what you have people do is literally take the cuts, stick them in the little cup. It goes in a little tube.
Are we supposed to talk? Are we supposed to be talking about?
Look, we can cut this out. We can cut this out.
No.
Okay. No, that was cool.
But I mean, it is really cool because
I think we might even have some film of it.
For those of us, look,
if not, we can get some.
For a long time, a lot of us have. It's been the Ziploc bag. Hey, everybody. Just wanted to remind you that if you want to try any of the products that you see us talking about or using in our own gardens, go to jugdealers.shop. And now back to the show. A lot of us have. It's been the Ziploc bag with a little bit of wet paper towel around the bottom of the stems.
Oh, man.
And you send it to the homie, or you're getting on a plane. I mean, look, this is just the way we've done it. It's not a freaking mystery. But the fact that you've kind of really turned it into this thing where it's unbelievable. And you get them, you've actually gotten them where they root in the cups and you just grow them out in the little cups. And you were talking about people doing, pheno hunting in the little cups. And I mean, you got it.
So we really, at the end of the day, like I said, I started as a grower and a breeder, and
so you still think like that? Yeah.
So I'm like, man,
I love that. I love that.
I wish I had this, and now I'm a scientist. So I'm like, I'm just going to,
I'm just going to make this shit. Well, let's talk a little bit more about just really tissue culture in general and kind of what it's doing, a little more on a molecular level, or maybe not even molecular, but on a single cell organism level, because reality is like you are in a sense affecting the plant's natural flora, right? I mean, it's kind of like your gut flora, right? If you just strip the whole thing out and it's nothing. So all the natural plus the unnatural, like now you're kind of a wide open palette, right? Yeah. So can you talk about that a little bit?
Yeah. So, you go through the process, especially meristem tip culture.
Yeah.
Right? You're stripping everything out.
Right.
There's no beneficials or pathogens or, anything.
Nada.
If it's done properly.
Right.
So
I mean, that is a big if, by the way, but yes, go on.
Yeah. So there's such a big correlation there with health and just survivability, right? In general, that we still have no idea. I know the technology is getting there so we could study it as far as the microbiome interactions with not only humans, but plants too, right? So if you take something that's completely sterile and you pull it out of culture, out of that sterile environment, nothing to compete with pathogens. So they'll just take over and you're back to square one again, right? So inoculating with beneficial microbes right off the bat. Like I don't sterilize cubes that I'm, pulling them out in. I inoculate and you see such a huge difference in performance.
Interesting.
So it's kind of a
very interesting.
Few different parts, right? You have the methylation piece we covered. You have the getting the right formula. And then you have the bacteria side of it.
Right. The floor, getting the right, that's all I can think of.
Do you have a favorite bacteria?
Oh man.
Oh wow.
We've
been starting to really dive down that rabbit hole. We got an episode on trichoderma coming out, and it's really, really interesting to me.
There's so many.
Well, not only is there so many, I think
bacillus is,
it's the interactions too. And I think that's what ultimately is what it comes down to. And it's same thing kind of with like hormones or really any of these things. It's always these ratios, and same with terpenes, right? It's these ratios. It's not all of one terpene or none of the other, right? And I think that's like the interesting thing about, nature and science is just that it is always never just this one thing. It's always that mix that, that it's always the beautiful mix. It's never, the entourage, right? Was the one coin term, which I don't think is bad.
Well, there's a better term out there. It's funny for us because we're always bashing hypochlorous acid. I don't love hypochlorous acid because I'm like, it kills all the good stuff, and here we are talking about a sterile environment. And the first thing you say is bust out some bacillus, in which, hey man, this is something we talk about all the time, is the importance of bacteria, mycorrhiza, all these different things. And, the fact that you're talking about the importance of it in tissue culture, it's beautiful.
It makes really total sense to me, but I did not know that. Around, that was not something I considered. Now, before we move on, because I do want to talk about genetic preservation as the next kind of piece of this, but have you ever had any like just straight horror story? Oh God, the entire lab is... because of do you have any like I don't know why I think that, but it's like it just seems like when you're on that kind of razor's edge. So yeah, what do you got, Jess?
I did actually kind of recently. I wouldn't say horror story.
Okay, all right.
I've been doing, like, when you guys came over, I've been doing a lot of work, man. Like,
right,
so many cultures, genetics coming in.
Sure.
So I ran out of media, so I made a fresh batch of media, and I make big batches. And just started using it right away because I didn't have any. So that's one that will catch up with you. And the batch of media got contaminated.
Pro tip.
First time it's ever happened to me because I got a new pump and everything, and I was like, oh man. So in that case, in a TC lab, it's a no-no. First of all, for everyone watching, don't do this. You want to incubate your media, make sure it's not contaminated before you use it, obviously. So I was like, what are these little specks in there? Oh man. So I had a new fresh bat. Well, it turned out my pH meter was broken, and my autoclave had to fix that. So it wasn't my fault.
Oh yeah, it was the equipment's fault.
It's equipment's fault. But in that case,
it's always more than one thing. I tell people it's always more. When it does catch up with you, it's
always compounding.
It's always three things. You're like, how did all this happen?
All at once, man. But yeah, I just had to go transfer them into fresh media real quick. Man.
Okay. So obviously,
not crazy. But.
Okay, so and obviously, but it's a heck of a reminder to stick within your SOPs, right? It's something like that. Absolutely. And even when you've been doing as long as you had, and you're kind of like whatever, I'm just gonna skip over this one little step. But I mean, I think that's also just for anybody out there, something to think about in your grow rooms is doing the little things right? Because to get to the big things, to get to your goals, you got to do the little steps along the way.
Make your bed.
Well, and or anytime it was like I wanted to skip that cleaning step, right?
Yeah.
But any time I in my personal stuff where I've skipped a step, it does catch up with you. So I think that is a great example.
And a quick pro tip from you that we saw in the lab that day. Why don't we use 99% alcohol in your cleaning
for just general cleaning?
Yeah, I think it's a great tip for anybody watching. When you said it to us, I was like, bro, that totally makes sense.
I did not know.
But why don't you tell people what you use and why you use it?
So 99% is good for getting sticky fingers and hash off scissors and all that, but 70% because 99% is it'll evaporate
very faster. Yep,
and it can actually kill. Microorganisms and whatever you're trying to kill, right? Sterilize,
right?
So yeah, always use 70%. And it's cheaper. I just buy 99% diluted. Right?
I was going to say, for those of you who don't know, you can just dilute it. You're going to dilute it with water.
And if anybody missed that,
30 mils.
And if anybody missed that, basically there needs to be enough contact time with the micro, with the stuff you're trying to clean off your hands, so that alcohol needs to stick around long enough to actually kill it. If it's too high a proof, like a 99, it's just going to evaporate too fast. So all that shit's still going to be on your hands. But I was like, I never thought about that. But
when you said it in the lab, I was like, bro, we got to mention that on the show for people, because I think a lot of people, quite frankly, waste a lot of money on 99% alcohol, thinking they're doing more than they are. And,
I'll tell you right now, this guy right here, I'll tell you right now.
And don't get me wrong, for cleaning pipes, for cleaning your hands, for cleaning all that stuff, it's great. It's great. But for cleaning the lab, tone it down a little bit.
Not to mention, it's also way more flammable. So.
Everybody saw that.
Okay, we will
go ahead, Grambo, chime in.
No comment.
Okay, all right. We're going to move into our next segment, which is something that a lot of people really talk about, and that is, saving genetics, long-term preservation of genetics. Like, in theory, forever. I mean, of course, we do have different places, like even in Fort Collins, where we have a seed preservation facility, right? A lot of it's done with liquid nitrogen and things like that. But obviously, they're employing some of this synthetic seed technology, I would think, and tissue culture technology for, various different economic crops and whatnot. So, I think general understanding about tissue culture is really two things, or at least that's what people think. It's cleaning genetics and it's preservation.
And keeping it. Yeah. How long can you keep a genetic, man?
I mean, indefinitely, really, right?
And okay,
do you have to keep like growing it out a little bit and like just putting it back in TC, or is there a way to just keep it in stasis?
So there's a lot of different, like I said, you want to picture tissue culture as a tool, right? There's a lot of things underneath just tissue culture, right?
Right.
I would say the most stable version of that, of genetic storage, would be cryogenic storage.
Okay.
Cold storage.
Cold storage, yep.
Right? You're slowing down the biological process. And
makes a lot of sense, yep.
Right? So if you're keeping it in vitro, so you have it under low lighting, lower temperatures, in a vessel, it's still kind of slowly growing. I don't like to keep them in there more than a year at a time, and I could pull them out and then just put them back in.
Right. Freshen everything up, new agar, fresh hormones, everything like that. And that's one of the ways you kind of would keep vigor and keep it from, and again, we're not trying to use the word drift, but from degrading, let's just say that.
Degrading, mutations, and that's just because I don't know yet. I want to dig deeper into testing and then feel confident.
Have you done
there forever?
Right. So you're comfortable at a year right now. Gotcha.
Have you done any type of testing where you've maybe taken 3 samples, taken one out in a year, and then let the other 2 ride for a little while longer? I mean, the thing about this is and I think we all need to understand as a group that like real testing and what we're all shooting for, this shit takes years, bro, years and years and years. And I've kind of sat with you and looked at your data. The amount of data that Jesse has is unbelievable. He's got pictures and family trees and every other thing you've seen. But the reality is, for you to collect this type of data, it takes years.
It takes years,
and I think that's something that's really hard. How long have we, I want to say the first time I saw a TC in cannabis was like maybe 2019, in which people were, when I tried to convince the guys at Curaleaf, they're like, oh, we're not doing that. It's too expensive. 2 years later, what do they have? They have tissue culture, and they're buying what?
Well, here's what's interesting though too. The ROI is not always there. I remember the classic story of them doing, like Hershey's actually looking into it for cocoa plants, and for whatever reason they just weren't, the traditional styles of propagation and whatnot worked as such that TC didn't work, right? But you talk about a higher value crop like orchids, or we've already talked about strawberries, but orchids, that's the same thing. You go to Hawaii once a year and you buy a few million, different orchids that all show up in vitro.
Yeah.
You know what I mean? So like for these high value crops and high dollar crops, obviously it is a big deal. And cannabis, I think, falls right into that, right? And both when you talk about variety too, because orchids, being the largest plant family in the world, it makes sense too, when you've got thousands of different varieties, right? And of course, they're a little bit easier to identify when they have all those different morphological differences. Here, we're really having to kind of go back and do the genetic markers and things like that as well to really definitively say that's in this line or that family tree. And again, we did did look at some of your spreadsheet stuff, which was absolutely extensive. I mean, you've done an incredible amount of work. And one of the things we talk about on the show is how science really is that reduction of variables, right? Mm-hmm. Which takes time.
Yeah,
I think I made the joke like I don't have any friends. I just do this all day.
You're easy to get a hold of because you're always home. You're not wrong there.
It's and that is frustrating. And then of course, so many times, with any of this stuff, it always comes down to strain specificity. Almost no matter what you're kind of talking about, it's hard to throw that softball right down the middle. Which I was actually somewhat surprised you kind of have been able to adopt a one size fits all kind of thing. I would think that'd be a heck of a lot easier in the way that I would want to go. I just, you always figure, there's so many differences. You know what I mean? There's got to be different for different strains. But anyway.
So I got a question for you. It's actually 2-part too. I know the one answer because you told me the story and I was like, holy fuck, that's crazy. But have you ever had a cut that you thought was dead come back from in the tissue culture bank? And how late, I think there's a lot of us who, in the process, we lose a clone or we lose something and we're here at the end and we realize we don't have any clones of it. And you're like, oh shit, I got to re-veg. What am I going to do? How late can you do tissue culture? And what, with that first question, I
gotta show you the picture.
What's the late, what have you ever brought something back from the dead?
So, absolutely. I wouldn't say the dead, right?
And look,
it has to be a
the brink. Yeah, when
you told me what 4 days you said it had been harvested 4 days, and you said, that's pretty dead to me, man. Like at that point, if I cut that plant down
4 days ago,
and I'm realizing like without you. There's no, it's not like I'm taking that bud and putting it back in the ground and going, oh God, please. Like, no, this is a Jesse. So this is where you're calling in the troops. So you're saying, in answer to the question, I could pretty much take tissue culture up till day of harvest.
So there's different types, right? So all this works on what's called totipotency, like a single cell has all the information.
You'll be able to differentiate into any cell.
Create an entire new plant.
Totipotency,
right?
I remember.
So with that, you can do root tip culture. I can pull some roots.
I was going to ask that one too. You brought it up. Awesome.
Pull some leaves off the plant, grow it back into a plant, callus, pretty much anything. If you have, so that's what I do have other formulas for. Is right. These different versions.
Of course. Sure.
Stuff, right? Sure. Rescue protocols. Cause that's like I'm running commercial facilities. Like we lost this cut.
I bet I think that would be like the bigger and bigger part of the business, honestly.
Yeah.
Yeah. It happens. So I get calls all the time. Like, man, can I bring in some flour? I'm like, yeah. All right, man, whatever. So,
yes, you can. Actually, you can.
You can. It does work, but as long as there's living cells there.
Right.
So, we had one in, I think it was about 4 days hanging in the dry room, and there's still stuff happening inside the plant.
Of course.
There, right?
Yeah. Water movement and whatnot. Yeah, of course. Water activity.
We got it to shoot. So the picture, I don't know if I sent it to you guys or not. I'll send it to you. It's like a dried, looking bud with a shoot, just
one little shoot
popping out the middle.
That is
amazing.
I mean, that is not only insane, but I feel like game changing.
Well, and do you Well, yeah, no. I was going to say, no, that's huge. Like, you want to talk rescue protocol. Please don't forget that, folks. Rescue.
If you know anybody else who you think is doing this, like I'd love to see it. Well, I ain't heard nothing like this before.
And before we move on to, I want to talk a little bit just about like long-term, obviously cryopreservation, things like that. Would you be doing that in vitro, or would you be doing that like live tissue? I mean, or would it be, depending on obviously what you had, but what would be completely ideal?
Completely ideal would be, do the meristem dissection first, or excision, right? So you're starting with clean genetics. I always recommend that first. And then ideal scenario, I, try to have as many genetic storage options as possible.
Sure.
Seed, Mother Nature knows how to store genetics.
Damn straight.
Right? Pollen, so we can preserve pollen for 10 plus years. Easy. Develop that.
Good to know.
Yeah, it makes breeding
males are important. Males get overlooked.
Yeah.
Males get overlooked, dude. Males are super important.
I put more effort into the males than females, honestly.
Interesting. It makes sense, actually.
On the tissue culture side, having long-term genetic storage. So they're in vessels, just kind of slow rolling, right?
Yep.
Lower temperatures, low stress environment. So we have actively growing clean tissue, and then cryo storage, because that's where you're going to run into less problems over longer periods of
And
time.
what would you put in a cryo? Like just a small piece of leaf tissue, or would it be actually like an in vitro plant? What would you actually put into that type of?
You could do a lot of different versions. I would stay away from cells or callus, right? So if you start with a leaf, you have to generate cells of callus and then turn that back into a plant. So there's a much higher rate of mutations and
You got more steps. Yeah.
But for breeding, that's what I want to do. Right? I want to see some just weird stuff happen. But having, clean tissue and you can get a bunch of different versions for this. So you actually have, looks like a regular plant morphology, that tissue, node tissue that's clean from, dissection.
Yep.
And you can store it that way. The cold storage versus cryo, that's a big difference.
Big difference. Yeah.
Right. So cryo, you can freeze anything.
Right.
Right. Bringing it back, is it going to be the same? Is it a leaf tissue? Is it that?
Yeah, that's what I was asking. That's what I was curious.
Things can get weird.
Yeah.
I would use cryogenic storage for DNA. Definitely DNA, and leaf tissue. So you can, extract more DNA.
Right.
Research down the road too.
Right.
And then everything else, like I would do, cold storage.
Sure.
So they're slow, really, really slowed down, and they're essentially in liquid media. And you can kind of have like a file, like in these bags that you can just file, like number A296B. Like there's Gabe's, he wants it back. Like bring it out.
Bring it out.
What is it going to take you on your side to, if I call you up, because we're going to go down this road in a little bit, but if I call you up, how long would it take if I said, hey, I need my genetics back, can you send me some cuts? How long does that take?
So it depends on like what stage it's at, right? A lot of people, they'll be like, hey, I'm going to get a new facility in 6 months. Can you get some stock up? I'm like, okay, cool. 6 months. Easy. If I have similar like crop rotation, you got a bedroom, you got enough moms growing at that stage, you can easily take a bunch of cuts, right?
Yeah.
But if you're keeping one mom around,
that's going to take a little while to propagate out. Yeah.
So same thing, just in vessels. So if it's long-term storage and you're like, I want this back and I got to pull it out of the freezer, that's going to be a lot longer.
Right. Yeah. So no, that makes sense. Kind of just what stage you have it at.
Yeah. If I have nodes, multiplication vessels going, I can get it out in a month and a half.
So I want
pretty damn quick. That's crazy.
I want to go down that road in a couple of minutes, but I want to start off with talking about something you showed us in the lab that we were just absolutely blown away by. And that was the synthetic seeds. So you were actually, and we'll give GRAMBO the video to download this, but you were basically taking these small, nodal cuttings, and then you were basically encapsulating them in a synthetic seed. So like a clear gel.
Kind of an agar type.
Yeah. And in which you had this this pen injector thing that literally was just like spitting out seeds. So why don't you talk to our customer or our listeners about, what they are and how they can be incorporated into cannabis? Cause this is something I thought was really cool, and it gives people the ability to really send you genetics and have you send them back, and they can literally just store them in the fridge until they're ready to take them out, so.
Yeah, there's a lot of stuff you can do, right? So what we were doing, I put some nodes in there, right? So we're encapsulating node in, it's like a nutrient gel matrix that has everything it needs just so it doesn't die, pretty much. You don't want it to grow out of there because then it could die.
Looks like a little boba with like
it's like a boba too.
It's a little boba with like a little piece of weed in it.
Yeah. So if you have a lab set up, I could, and we worked on this so we could store effectively and think shipping.
Ship effectively too. Yeah. That's what you're saying. That was cool.
I was really intrigued by was just being able to ship these little vials of seeds.
Well, and that to me, that's one of those things that hugely will change the game for breeders, nurseries, MSOs, anybody that's trying to go to scale, get to scale quickly with clean genetics, right?
Yep. So if you have clean stock, and I can do those, I can do them fast.
Yeah, you were like,
so then I could send, something this big. I could send hundreds and hundreds, thousands.
Little fish eggs with weed in them.
Fish eggs. So a true synthetic seed actually has it's a synthetic embryo.
Right. So this is synthetic seed. This isn't synthetic seed.
I guess,
this is your kind of version of it a little bit. Yeah.
We can do both, but with an embryo, you can actually get root and shoots just like a seed.
Sure.
Right. So picture planting, sour diesel that's true to type.
Right.
Basically a clone from seed form.
Yeah.
Does it make sense though?
There you go. There's a shot of it, folks. Yep. There's a shot.
Oh, those things are awesome, bro. I was so blown away.
And he gave them to me in a little, like one of those little seed vials. So it's like 10 of them in there, just bloop, 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 bloop. Super cool.
Do you think this is tech the big places are going to be chasing down? Like, do you think it's going to change the way a lot of them do the game, or you think it's going to,
it depending on the application, right? For us, like I build labs and train and set up other labs. So it's like, here's all of your stock in a tiny package. They can start propagating in their TC lab on the other side. So it's really easy to send.
Yeah. That's what I thought was amazing. Stable. Yeah. That's what I thought was really cool is, if they have it on their side, they can pretty much have everything ready to go. So
Grandma, can you move that up a little bit?
Yeah. So depends on the application, and this stuff's been around for a while too. They're not difficult to make. What we worked on is the formula just for viability over time, right? So we've dialed it into where I can keep it in a refrigerator for about a year and get about 97%.
Well, and so that kind of do you think that has the ability to these type of synthetic seeds disrupt the current seed and clone industry? I mean, like on one hand, I could see how it could. On the other hand, again, going back to even, Mother Nature has created a really pretty awesome storage vessel. It's called a seed, man.
That's it.
It's got that hard coat on there. You know what I mean? It's like, it's pretty darn cool, right? So in some ways, do you just look at this as really just another option? This isn't something, another tool.
Another tool.
Yeah. That's another tool. So this isn't something that's...
Hey, everybody. Just wanted to remind you that if you want to support us, go to jugdealers.shop. There you can find any of the products that we talk about and use in our own gardens. And that's straight from us. So now back to the show.
Another tool.
Yeah.
That's another tool. So this isn't something that whether you're looking to or even you see it as a possibility to knock it off, because it's really just another arrow in the quiver, another tool.
Yep. That's it, man. And a lot of the stuff, setting up a lab, tissue culture is a great start. You can do a ton of stuff just with that equipment and that setup, right?
Right, because some of this equipment is basically, it's scalpels and it's autoclaves and it's the shit that you have in a lab.
You had that outdoor flow hood. Like, I was like, what? Like, if you really looked at Jesse's setup when we were out at Dude Gross Cup, like, he had like this box, and if you're not really thinking about it, you would just think it's just, some normal box. But that thing was a flow hood.
That's it.
Well, and it's, and that's what it's got to do. It's got to be moving the air in the right direction. So we're moving anything that could be bad coming in there, right?
By
so
definition.
It's flowing the air out of there. Yep. So you do need a little bit of equipment, and obviously you were able to acquire some very high quality equipment, so that you could actually get a legit lab started, which can't be the easiest thing to do for people without just having a bunch of money, right? I mean, there's a certain entry.
I love that, Gabe, because when the market in Colorado crashed, right? The research gets cut first.
Unfortunately,
we're spending all the money. So I just chose to keep equipment and keep going.
Which I thought was really cool. You said that right off the bat, because it's kind of like, gee, where'd you go? So he's like, well, this shit went downhill, and I just kept the equipment and moved on. But it's the fact that you had gained that knowledge, that experience, and then to be able to really roll with the equipment and say, well, I know that I can create value with this stuff. And that's obviously one of the things that you're is one of the genesis and the whole points of the company.
Yeah, and what we're trying to show people is like it's not that hard,
right?
We've done a lot of the hard work. Like,
you're not trying to get formulas.
And we're going to be working on video content we were chatting about earlier. Like, here's how to do everything,
right?
And we're starting to film stuff, and I have SOPs, formulas, everything, because I see from my grower breeder perspective. It's like, man, I wish somebody taught me this a long time ago.
Well, and shout out to all the people that came up to us at DGC. They were definitely, that got quoted over and over to us. It was like, thank you for saving so much time. Thank you for not gatekeeping the information. And I think you're doing the same thing that we're trying to do, just with, a different set of information. So, I really, really do, I appreciate you doing that, because I think a lot of this stuff, has been held back for a long time. You know what I mean? So, but why don't you give people a little bit of an idea of what the heck it's like being in the lab, being a real scientist in your backyard? Can you paint a little bit of a picture for us? You get up, you turn the lights on in the lab. What's the daily grind like?
Oh, it's just like cultivation, right? Schedule.
Yeah.
What am I doing today? Because, what people see is the TC. They're like, oh, look at these vessels and plants and things. But really what it is like, you have to be tight scheduling.
Yep.
QA, QC, so quality control, a lot of that stuff, SOP development. All of that. It's just a lot of management, but the same stuff is for data collection, right?
Yeah.
Building in, so everything's easy, automated.
Yep.
So that's a lot of where I start my morning. Where it gets out of control is because it's in my backyard. I'm like, think of an experiment. Going to the lab.
Yeah. It's great. It's great and bad at the same time. I mean, and I guess, as we're kind of getting towards the end here, one of the things I always like to, open up for anybody, any common misunderstandings. Do you think there's something that, that you've heard over and over again, people talk about with tissue culture that just is complete bullshit?
Oh, there's a lot.
Where do you start?
There's a lot. And breeding too, man. I think the biggest one is it doesn't take $10 million to build a lab. Right?
Okay.
You can do it for cheap or cheapish, right? You can build your own equipment if you want to.
Mm-hmm.
And it doesn't take a PhD to, successfully do it. Right. So that's something we're trying to help with and just share that information, because that's a lot of the first things I heard when, talking to people. It's like, oh, it costs $10 million. I'm like, what?
When, what, where, how?
Yeah. And the training or really doing it, I think that's because I've trained a lot of people at this point. It's putting the time in, because once you're not uncomfortable working in a flow hood and handling your tools, it's not that hard.
Well, and that was one of the things that I saw, and I picked up right away, and Gabe did too. But it's kind of like being a mechanic, it's like, hand me, a wrench, and it's like, I don't use it every day. I'm clumsier with it. You put it in, and a guy uses his hands every day, and it's like, oh, fuck, he makes that look easy.
Well, and the big thing I noticed with you is, just like when I was a mason, I had really specialized trowels for doing certain things.
Yep.
I mean, you're using dental tools. You're using.
There's a whole.
There were a couple. For me, who's like a weird tool guy, like I've always just been into weird tools. You had some really neat little scissors and stuff like that.
The tiniest.
Just working at really small scale, and it's like, some of us get excited about the smallest shit. But that was the kind of thing where I saw you using. I'm like, man, he's using a this bent way dental tool. I'm like, that's fucking great.
Right.
The right tool for the job. And some people hate them.
No, you were
tools. And
you were like a surgeon. You were like a surgeon in there with it, and I was like really surprised because I'm watching you work on the screen. And then you look down on the glass plate, and you like to use. I asked you why, but you actually like to use a really thick piece of glass, it's like a 3/4 inch, 1 inch thick piece of glass.
One of my favorite things to do is you go to like Goodwill or home stores and be like, I can use that for TC. It's just
I just finding random shit to look for TC.
That's perfect. For
Josh, can you move it up a bit? Yeah, we'll go all the way to 6. Yeah, there you go. As far as just kind of looking ahead at really where, a lot of this stuff is going, like what really excites you, and where do you think basically cannabis biotech, because you truly being someone that I think Gabe and I really look at as being on the front end of this stuff. What do you think next 3 to 5 years?
Yeah, we'll be doing a lot of research as well, because we've created these tools, not just TC, but we've created, we have tons of other stuff that's exciting. Distribution, because we're working with breeders. We have,
and that's distribution of genetics, correct?
Correct. Yeah. From TC. Sure. So we have our partners in Florida, they're set to I mean, they can produce like 5 to 9 million plants a year.
So we're talking production. We're talking actually at scale, clean production.
Yeah, and this is awesome. We were excited about talking with.
And we've talked. This has been talked about for a while, hasn't?
Yeah. Well, it's funny because I had said to Jesse, 11 years ago when I met Fletcher, we had discussed. He said one day people are going to be getting it all. It's all just going to be. So people are just going to go to the nursery and they're going to buy this shit. And I was like, Fletch, you're out of your freaking mind. And yep. Now I'm seeing it coming around. And I've said it many, many times. There's a lot of stuff that Fletcher has schooled me on where it was like, I was like, no, Fletcher, you're crazy. And now it's like 10, 12 years later. And it's like, man, Fletcher's right about all this. But we actually talked about it. And I've been putting it out to people to be able to take your genetics, send them to you, have you clean them up. And then you send them back in a sealed container. I'll give GRAMBO the video, but in a sealed container, their own genetics, ready to go, no pests, no pathogens, everything. And to me, not only is that the way to start a facility, but then you raise the question: Are we going to need moms anymore? Are we, is it going to be the type of situation where people are just calling you up? And
no. I'll take that a step further. Do we need bedrooms anymore?
Yeah. Well, I think,
you start thinking about I
think if you're going straight to, I have my own opinions about Sea Green, and I know Billy Boy and I have gone back and forth about it, and look, it kicks ass. So if you're doing Sea Green, do you need a bedroom at all? Or do you just,
I'm just posing questions.
Yeah. And the reality is, we've said this on the show before. We're the only crop that splits ours into cloned veg flower times.
Right.
Yeah. If you look at people who do agriculture for a living, it's a 90-day crop. It's a 100-day crop.
And that was kind of my point too. Is there a middleman that pops up
in which
it takes it from you to then be the veg guy?
But
I'm just,
I've talked to you about it, in which, I think something that, we're low key working on, and you definitely kind of got my brain going, is have someone who has a subscription and clones literally just your genetics, show up every 2 weeks. They're ready to go. You've gotten rid of your clone room. Yeah. You've gotten rid of your mom room. Yeah. And now you have stuff that every time you get it.
Specialization too, right? You're not having to do so many things under one roof.
And
overtraining staff, all these things, right?
And for the longest time, that would have been unbelievable. But now that you've kind of presented,
it's totally believable.
Now that you've presented the option where I can use my own genetics, we can use Jerin's Gorilla Glue, we can use his GMO, and we know it's ours every time. For me, it's game changing. Do you see it going that way?
I do. I mean, in a lot of ways, right? So one of the reasons we partnered with our friends in Florida is to help breeders, help cultivators. Right. There's other models out there that are just terrible. I've seen the contracts, and I'm like, nobody
wins.
Afford this. Yeah, nobody wins. You're locked in, tons of feet. So we're trying to be, the mothership labs will be the beginning of this pipeline to where that's just another layer. I can protect things from going to the big nursery, and we could sell mom stock
And you've talked about pairing up with breeders, actually having some type of marker that puts them with the genetic. And then if they want to keep the genetic, they can just keep the genetic. It stays banked and they're the only ones who get it. But if they want to sell said genetic, then you've actually made it so that you can pretty much, because I know I have a lot of buddies, including myself, including Jayhawk, we've held these genetics for a while. We know what they're worth and we like to get rid of them. But you actually give us a clean avenue where we can guarantee them to people to be clean, sterile, ready to go.
Yeah. So that'd be for Colorado. We're getting, finishing up, looking at a new facility so we can expand. Get some techs in, start collaboration stuff. And Corporate J, we call him Corporate J. He's keeping it moving.
He is.
Yeah, that's what you got to do.
He's the man. He keeps me in the lab and keeps all the business development stuff rolling. But then we could do exactly that, right? If you work with us and you're a breeder, you don't have to keep all these moms. We could sell them for you. We could work it out to where it works. I don't even have to know what they are. And that's the one thing we're still, and I'm very open just to talk about is like, how can we improve this model because there's so much trust and things.
Right. And trust has been breached. Right. And so that's, and I love it how you're still saying it's open. It's a work in progress. And I kind of tried to say at the beginning too.
Always will be.
Yeah. Right? And you're like, hey man, we're evolving. And I think that's a great thing. Even like saying, hey, look, it's, this is, J1, this is number 1 genetic from Jerin. I mean, there's a bunch of ways that things could be cataloged. And I think people have to keep that in mind as well. One thing I wanted to touch on super fast.
The tech.
Yeah, it was tech, man. Automation.
We talked about this in the shop.
Yeah. Where's this shit going, man?
Well, when you and me and Ryan talked about it, and and you were like, bro, there's going to be robots and like.
Oh yeah, so we are
robots are definitely.
I'm like, you really think so? And you're like, bro, it's totally robots.
I already have some picked out, right?
Names and everything, dude. Oh shit!
Yeah.
So there's a lot of different types of robots.
Y'all better get your shit together because the robots are coming.
Good point. Robot is a loose term, somewhat loose term.
Specifically,
not necessarily
TC,
sentient thing. Yeah.
Yep.
Right. There's some for TC that are incredible.
Right.
Takes a lot of workload off my shoulders.
You and I were talking about a defoliating robot too.
Defoliate? Oh.
Well, man. I saw, in Essen, Germany, these little explants getting, these little robot arms planting up trays, 220 cell trays. I mean, with an insane amount of precision, you know what I mean? And they were taking all the cuts off of there, putting them on a belt and then putting I mean, it's, like you say, it's definitely, part of the deal. And then I do see I see an evolution where you do skip mother rooms and you do skip things. And again, with a high value plant like this, when you can have bank genetics and it really makes sense to maximize your growth space and also specialize in what you're good at. Some people are great at vegging. It's why you do specialization under your own roof. Yeah. Why wouldn't you specialization? Why wouldn't you specialize your operation in general?
When I have a clone manager, that person gets paid a decent amount of money. So if I can replace that, and I'm not being like that,
but for real,
I say it to people all the time, and this is an unfortunate thing, but the truth of the matter is labor is your highest cost. Yeah, and it doesn't matter what you're doing. So if I can cut labor and as I love to say, not for nothing, if I can on top of it ensure that I am getting great genetics every time. I can't tell you how many times I've walked into a clone room where a guy left the dome off too long, and all of a sudden I'm having to friggin' shift the whole product plan because I don't have any face off. Bird, you know exactly what you did, Bird. But I don't have any face off because you left the dome off for an hour and I told you to leave it off for 15 minutes. And with what you give people, it's literally a peel and play.
Yeah, you peel it off, bottom up, ready to go.
So that, I mean, just in that respect, to have that peace of mind is a beautiful, beautiful thing.
The one thing too, and I know that we promised this, but I wanted to get into this before we got out here, and I know we've kept you guys around for a good one today. I wanted the one big thing we talked about this too, just the practicality of, really truly outside of an autoclave, and maybe the answer is just an autoclave, but how do you truly clean your instruments specifically for hop latent or really any viruses of that nature, right? Because we've all heard, hey, alcohol is not enough. Mm-hmm. Jesse, can you, I wanted this is something everybody, I want y'all to listen up.
You got that banging little unit.
Go ahead.
Too, bro.
Oh yeah. Mine talks to me. It sounds like the voice from Resident Evil. It's crazy. Just kill everything with fire.
Yeah. So really autoclave is
when in doubt, autoclave it out.
Yeah. Cause it's steam and pressure. Right. Mm-hmm. And then bleach works.
Okay.
Right.
What percent are you talking? 20%?
10%, I believe, will kill.
Okay. 10% bleach. Okay.
Yeah,
okay.
Sterilized tissue.
10 mils.
I think it's like 10 minutes.
10 minutes. So you'd actually leave it in there for 10 minutes and 10% bleach.
Yep.
You hear that, folks? 10 minutes, 10% bleach, put your instruments in there, let them soak. And then obviously either have individual, instruments for different plants or different genetics, or take the time to, do one whole plant and then soak that shit for 10 minutes and then go on to the next plant. So that was something I really,
isolating,
right.
That's the key. And if somebody has viral, hemp latent viral in their facility, just strategy, we could flush it out, but not if you're just walking around.
You're all willy-nilly all over the place. It ain't going to work. Yep. I guess, is there any last things that you want to say? I mean, really, we've gone around the map. This has been a pleasure, and there's definitely been a few things that have that I've learned and some myths that have been dispelled, and obviously just giving people an idea of where some of this stuff is going. But remind people where to find you, and also, if there's any last and parting words, this is your moment.
Yeah, check us out, the Mothership Labs.
There it is.
Instagram is low-key high tech. I spent my whole life trying to make sure people didn't know.
Yeah. Well, trust us. But we've been through this ourselves.
Meanwhile, I'm out telling every single person I know, you got to hook up with this guy. So the reality is, after this episode drops and 10,000 people see it, they're going to be like, "Oh, this is the guy to get in touch." I mean, I can't tell you how many people I sent over your booth just to DGC.
Oh, we appreciate it, man.
And it's funny because there were a lot of people at DGC. We had a guy that we talked to about the Death Star and stuff, and there were a lot of people at DGC.
You know who you are.
That were like, "Hey, I've had this genetic 10, 20 years. I don't know what I got to do." And I'm like, "Hey, go over to Jesse's booth. Well, and Jesse will take care of you."
I'm so excited that we do have more of these opportunities available. I guess, is there something too that you would advise people to look into testing stuff, or you figure, Jesse, what's the point? Just get it cleaned up because there's going to be some shit in there. I mean, what would you say?
I always get it tested.
Okay.
I always go to TwoMe Genomics.
Right. Out of Fort Collins, Colorado, I believe.
Awesome.
Yep.
Man.
Okay. There you go. And they have different viral testing kits, right?
Yep. And we'll be getting
maybe you could pull that up
and stuff too. Because if you got it, your other plants do too.
Right. So you're using it as an indicator and then saying, and don't think it's just one, like they're all going to have it. Yeah. What is it? Is it TOO? How do you spell that to me?
TUMI.
TUMI. That's right.
Yeah. I mean, I would encourage anybody who's had genetics a long time, reach out to Jesse.
There they are.
Give him
TwoMe
a
Genetics.
Chat, tell you what he can do for you. Tell him the Jug Dealers sent you.
Tell him the Jug
Dealers. Hey, we heard about you on the Jug Dealers. What can you do for me? I got this 20-year-old Skywalker, or SFV.
Right. I mean, look, this one went. We're test tube to terps on this one. This was really interesting, man.
That's the truth.
So thank you guys for all coming out. Thank you so much, Jesse, for coming to hang out, spend a little time in the Jug Den. If you guys do have any questions, you know where to find us. Hit us up, direct message us, hit us up on the Instagram, hit up GRAMBO. Comments on the page, whatever. If you guys got some specific questions, who knows? We maybe even put a whole nother episode out based on some of the questions you guys have.
And I will say, I'm the one that's always running the Instagram. People will send me very specific questions like, how do I insert this? And I go, I'm me. So hit up Jerin or Gabe, Gmuneyluv, or at Jerin H.
At Jerin_H. You are correct. GRAMBO. He is the man behind the glass. He's definitely a big part of all this. You guys know who he is. The man, the myth. Jesse, thank you again for coming out. Till next time, guys.
Me, guys. I really appreciate it. And no matter how much science we want to use, right? If it ends up tasting like shit, doesn't matter.
You're not wrong there.
Right?
You're not wrong.
It still is connected to the people.
Hey, man, like I say, especially with this plant, there's always a mix of art and science. And I think that's one of the most exciting things and fun things about it. So till next time, guys, from the Jug Den, peace out.
Peace. Thank you.